Okay, this is our third and final installment of our lecture number 1 on the vision system. So we're going to do an introduction to the retina and the photoreceptors. We're going to learn a bit more in detail about them in lecture 10, but this is something to get us started. So the retina, you'll recall, is that inner layer of the eyeball, covers the entire inner surface of the eyeball. And this image that we're seeing here is what you can see when the doctor uses their ophthalmoscope and gets really, really, really close to you and looks inside your eye, because of course the eye is filled with clear things, and so you can actually look and visually see. So this is a photograph that was taken basically from one of those ophthalmoscopes. So there are a couple of things that we see when we look at someone's retina. And one of the most obvious things is the optic disc. So it's circled and labeled for you here in this image. So it's this kind of round, bright looking area. And what that is the beginning of the optic nerve as it's about to head out of the eyeball. And as you know, nerves are bundles of axons. And so at the genesis, the origin here of the optic nerve, it's all just axons from neurons in the eyeball that are traveling then out to the brain. And there's no photoreceptors then on this part of the retina. So the interesting thing is that any light rays that land on this part of your retina, you cannot perceive them. So this is a blind spot. We have one in each eye, but they are in different places in what we would call our visual field. So because of that, your brain just seamlessly fills in information with what it's getting from the other eyeball, so you don't notice that you have these blind spots. But we will find our blind spots in lab on Thursday. So fun stuff. This is an image showing you the same thing. So let's see, so the red dotted line is the one we're interested in here. So if there's an object here and its light rays are coming into your eye in such a way that they are landing on your blind spot, which is where your optic nerve begins, you cannot see it. Now, of course, if it's an object over there, that means that the light rays coming into your other eye are going to hit a different part of your retina. So you can see it with your other eye, which is why you don't notice that you actually have these blind spots. So we have these blind spots, one on each eye, because the optic disc is located a little bit toward the nose side of your retina. It's for objects that are kind of out to the sides. Okay, kind of cool. The other thing that I want to point out to you about the retina is, and actually before I get here, notice there are blood vessels, right? So every part of our body needs blood vessels. You're like, wait, I thought blood vessels were in the choroid. Yes, they are, but we also have blood vessels that travel through the retina. It is a very active part of our body, so it needs a lot of glucose and oxygen. Okay, so that said, now we're going to focus on the macula and fovea. So the macula is this region over here, fairly circular. And then the fovea is right in the center of it. So the macula is the part of our retina that has the most densely concentrated photoreceptors. So we have lots and lots of photoreceptors in this region, and most especially in the fovea. So it's designed so that whatever you are looking directly at, those light rays of the object that you are focusing right on are hitting your fovea. And so because you have so many photoreceptors there, this is where you have the sharpest sense of vision at all. So it's kind of like on a screen, the more pixels you have, the more definition you get. It's the same way. The more photoreceptors you have, the more definition you can get, the more precise information you can get, and therefore the clearer, more detailed, the crisper the vision. So do me a favor and look at one of the words on this slide. Now, move your eye like over to, I don't know, the diagram of the retina, and out of your peripheral vision, out of the side of your eye, try to read that word. If you don't cheat, you will find that you cannot read it, right? So only in the very center of our visual field, only directly where we are looking, do we get that very high resolution, crisp vision. Okay, so this is that same thing. So just showing you a different diagram. So whatever you are looking directly at, their yellow doesn't show up very well. So if I'm looking straight on, dead on an object that's right here, those light rays are coming in and they're going boom right on the fovea. So I get the most information about whatever I'm looking directly at. Okay, so that's why you look directly at something if you want to see it clearly. There is a condition called macular degeneration. To degenerate means to kind of break down and not function, right? Now, this is a condition that can develop usually with age. It's usually an age-associated thing. Doesn't happen in everybody. There are risk factors and also lots of unknown factors. And there are different types. But what I want you to understand is if you have degeneration of the macula, so this is an image, this is a photograph of someone's retina who has macular degeneration. And you can see they've highlighted the macula with a green circle for you. And there's all these kind of white spots and there's dark spots, right? There's badness happening in their macula. But the macula is where you have all of those photoreceptors to see precise things. And it's also where you are looking directly at something. So in this image, let's imagine this is someone's grandchild, this little girl that we're seeing. For someone with macular degeneration, if they were looking at her, it would look more like this, right? So as you can imagine, this is very disabling. It can be hard to see because you can't see what you're looking directly at, and you also can't see with that precision and focus that we need to be able to discern things. You cannot read once macular degeneration progresses to a certain point. And of course, it would be really difficult, if not impossible, and certainly unsafe to drive. So this can be a very disabling condition. There aren't good treatments for it, although there's a few promising ones for the rarer types of macular degeneration. So it's something to be aware of. So retina, we talked about the optic disc, which is the beginning of the optic nerve. That is our blind spot. And we talked about macula and fovea, which is where we have the highest concentration of photoreceptors. So now let's talk about those photoreceptors. So they come in two types. They come in rods and cones, so named because of the shapes of the kind of the tops of these cells, the way they are positioned in this image. So let's talk about them one by one. Rods see in black and white. They're like light is present, light is absent. That's it. What's nice about rods is that they're very sensitive, so they can detect even low levels of light. So this is for our night vision, right? So in low light conditions, rods are helpful. But rods don't give us high resolution, precise information. We'll talk about why that is in lecture 10. So you get kind of a fuzzy image. So this is why at night in the dark, things look a little fuzzy, things look a little blurry. You can't read in the dark, and I'm talking like dark, not like dim. Okay, so that's rods. And then cones come in three different types because we can see in color. So they come in red, blue, and green, basically. Those are the three wavelengths of light that they respond to. So this is very much like if you've ever adjusted color on your television screen. You know, you dial up or down the blue, the green, and the red. And somehow between those three, you are able to produce all of the colors of the spectrum. Now, cones only work in bright light, so they aren't as sensitive as the rods, which is also why in lower light conditions you cannot distinguish colors as easily, but they are high resolution, so we get clear, detailed images. So then you might be wondering, do we have lots of cones in the fovea? Yes, right? So remember that fovea, the center of the macula, where we have our most precise, clearest, highest resolution images. And so what we're looking at in this graph, which is from the LIM textbook, is if I was looking at a section of my retina in my eyeball, and this is the side over towards my temple, and this is the side that's closer I need two hands to do this, to my nose, right? So your fovea happens to be located kind of midway, but a little bit more out to the temporal region. And look at, so this tells you how many of the different types of cells you have, what the concentration of cells per area is. Look at all of these cones. We have a gazillion cones in the fovea. So that gives us that really high resolution that we've been talking about. We also see in color there, and it works best in bright light conditions. In the rest of the retina, we have far more rods than we have cones, right? So the gray here, these are the rods. Obviously, as we kind of get out toward the periphery, we don't have as many photoreceptors, right? That's just peripheral vision. It's not as important. But look at all of the rods that we have and how much they outnumber the cones. So they only see in black and white, but they're really good in low light conditions. And this is why also not only are colors less vivid in low light conditions, but colors are also less vivid in your peripheral vision. But we don't tend to notice that because the brain just assumes and kind of fills it in. So we don't usually notice that. So one really interesting thing, the next time you're in a very dark room, like a very dark room, but you can still see a little, right? It's not completely dark. It's not a total blackout. You can still see a little bit. If you need to see something as best as possible, as precisely as possible, if you look just to the side of it, you will actually get a better image than if you look directly at it. Because if you look directly at it, all you've got are cones, there's no rods there. So you have to look a little bit off center, so it's a little bit out of the side of your eye, and then you'll be able to see it better. So go into a closet or, try it out. It's kind of cool. All right, so for this introduction to retina and photoreceptors, I'm going to start with photoreceptors. So the rods give us our black and white vision. They're a little bit fuzzy, but they're very good in low light, so that's our night vision. Cones see in color. It's pretty sharp, but they work best with bright light. And of course, they are most concentrated in the fovea, which is why we get such sharp, detailed images from what we're looking directly at. So that optic disc, remember, is the beginning of the optic nerve that we can see on the inside of the retina. There's no photoreceptors there, so we do have a blind spot, but it's in different places in our visual field for each eye, so we don't notice it. And then, of course, the macula and fovea, those areas of the highest concentrations of photoreceptors, particularly the cones, and that condition of macular degeneration, which can be very disabling because you are losing the majority of your vision and that central and detailed vision. Okay, so now you're ready for lecture 10. We'll get into the nitty-gritty of what's happening with the neurophysiology and brain pathways. See you there.